Today, I'll, I'll try to explain Mendel's experiment on hybridization and monohybrid cross. Myself, Dr. Ichhapurak, retired university professor in botany from Ranchi Women's College, Ranchi. Now, in this lecture, a brief introduction about Gregor John Mendel will be given. What is hybridization? What were the reasons for selecting sweet pea as experimental plant? Alternative forms in sweet pea, steps in hybridization experiment. What is monohybrid cross? What are the results of the experiment? Genetic expression of monohybrid cross. Mendel's conclusion. Mendel's first law, that is law of segregation. Now, a brief introduction about Gregor John Mendel. He was born in 1822 at Austria in a farmer's family. He continued his higher education at Brunn. In 1854, was appointed a science teacher, and he lived in a house in the premises of church. He used to spare free time in gardening and became interested to know about transmission of characters over generations. Now he started his experiment on sweet pea, that is Pisum sativum, in 1857, and he continued till 1865, that is nearly for 8 years. In 1865, he presented his paper before Natural History Society, Brunn. In 1866, the paper was published in the Proceeding of the Society, but nobody paid due attention to his work, as Mendel was not a biologist. He was simply a mathematician, and by profession was a priest. In 1900, Mendel's work was rediscovered by Hugo de Vries from Holland, Karl Correns from Germany, and E.V. Sharma from Austria. These three scientists named the laws of inheritance as Mendel's laws of inheritance in honor of Mendel. Or since then only, he is known as father of genetics. Although in his life he didn't heard anything about his work. He passed away in 1884. Only after 16 years his work was rediscovered, and they named the laws of inheritance as Mendel's laws of inheritance. Now, what were the reasons for selecting sweet pea, Pisum sativa, as experimental plant? Now, this slide shows sweet pea. You can see the flower is very large, and the corolla completely close the stamens and gynoecium. So, it is a self-pollinating plant. No, Pisum sativum is a self-pollinating plant. It is suitable for artificial cross-pollination. Bears large flowers. Emasculation and cross-pollination are easy. It is annual, so the results are obtained yearly. Pure lines are easily available as it is naturally self-pollinated. Means there is no chances of mixing up. It has several varieties showing contrasting form of characters. These are the characters or traits on which Mendel worked, like seed shape is the trait, and it has two alternative forms, round and wrinkled. In the same way, seed color, the color of mature seed may be yellow or green. Flower color may be purple or white. Pod shape may be inflated or constricted. The pod color may be green or yellow. Flower position may be axial or terminal, and the height of the plant, if it is tall, it is about 2 meters, and if it is dwarf, it is about 0.5 meter. Now, we can see in the table that in the third column, whatever is written are the common alternative forms and are dominant, whereas in the fourth column are the recessive alternative forms and are less common. Now, these are the diagrams of the same traits and their alternative forms. And these are again, flower position is axial or terminal, and stem length may be tall or dwarf. Now, what is hybridization in plant? Hybridization is frequently employed in agriculture to make stronger and healthier plants with desirable characters. In plants, hybridization is done by artificial cross pollination. For example, a cross between parent 1, which is tall, with the genetic makeup or genotype capital T, and parent 2 or dwarf with genotype small t is expected to produce progeny with genetic makeup capital T small t, which is a hybrid or the first hybrid or filial generation or F1 generation. Hybridization was the basis of Mendel's historic experiment with garden pea. Inheritance studies require crossing plants with contrasting or alternative traits by considering one plant as male and other as female, although both parents are bisexual plants. Both the parents have male as well as female reproductive parts, but the parent whose pollen grains are used is considered as tall as male plant, and whose stigma is used is considered as female plant. Now you can, this slide helps in understanding how hybridization is done. Now as pea plant is naturally self-pollinated, it can be easily used for artificial cross fertilization or hybridization. In emasculation before anthesis is followed by dusting stigma or carpel with pollen from other flower. Now in the first step, you can see that there are two color of flower colors are of two types, purple flower and white flower. The purple flowered plants carpel is being used, and the white flower plants pollen grains are being used. Now in the first step, from the purple flower stamens were removed, and in the second step, pollen from stamens of white flower were transferred to carpel of purple flower. In the third step, pollinated carpel were left to mature into pod. And in the fourth step, the planted seeds from pods. The plants were left to form seeds, and then seeds were grown in the next generation to form plants of F1 generation. Now, what is monohybrid cross? Monohybrid cross is a cross when only one trait and its two alternative forms are considered during hybridization experiment. Tall male plant is crossed with dwarf female plant, or in another monohybrid cross, purple flowered male plant is crossed with white flowered plant or vice versa. The steps in monohybrid cross, as for example, for the trait height of the plant, that is tall versus dwarf, first step is testing of parental plants for purity. The plants are tested for 2 or 3 generations, and if a tall plant gives tall plant for 2 or 3 generations, then it can be called as pure tall. Consideration of a plant as male parent whose pollen are used. Consideration of a plant as female parent whose stigma is used. From flowers of female plants, anthers are removed before enthesis or emasculation. So the main steps in hybridization experiment are selection of pure breeding plants. That is followed by emasculation. Then emasculated flower is bagged or bagging is done to avoid unwanted cross pollination. Then the seed setting, collection of seeds, then raising of F1 generation plants. F1 plants are self-pollinated. Seed setting of F1 plants, collection of seeds from F1 plants, raising of F2 generation plants. In the same way, every step is in the same way. F3 plants are raised, F4 plants are raised, F5 plants are raised up to F7. Then no. Emasculated flowers were covered in porous paper bags to avoid undesired cross pollination. Hybridization or artificial cross pollination was done by dusting pollen from flowers of male plant on stigma of flowers on female plant. Fertilized flowers were left on plants to set seeds. Seeds were collected separately from each plant. Then the seeds were sown next year to raise F1 plants. Now the F1 plants were left to self-pollination. No cross-pollination again. Seeds were collected separately from each plant of F1 generation. Then the seeds collected from F1 generation plants were sown to raise F2 plants. F2 generation plants were left to self-pollinate again, and seeds of F2 plants were collected to raise F3 generation. In this way, up to F7 plants were raised. Now, what were the results of the experiment? Monohybrid cross between tall male plant and female dwarf plant and vice versa in both the cases. In F1 generation, all plants were tall. Dwarfness disappeared. Now, what happened to dwarfness? Mendel was surprised what happened to dwarfness. F2 generation showed tall and dwarf plants both appeared in ratio approximately 3 to 1. In F3 generation, of the tall plants of F2, some plants gave tall and some segregated again into tall and dwarf in 3 to 1 ratio, and dwarf plants gave only dwarf plants. F4 generation, some tall plants of F3 generation gave tall only, and some again segregated in 3 to 1 ratio. Such monohybrid crosses were made for all the 7 traits and their alternative forms. And in all cases, the dominant form appeared in F1 generation. In F2, both dominant and recessive appeared in 3 to 1 ratio. Now, this slide shows the genetic expression and analysis of results of monohybrid cross. You can see the parent tall male plant is represented by capital T, it is homozygous. In the same way, dwarf female is small t, it is again homozygous. When the two are crossed, tall male plant will give gamete capital T and female plant will give small t, and on fertilization it will form capital T in the zygote, which will set the seed. And when the seeds will be sown, F1 plants will be formed. All the F1 plants were tall, although it was having the allele small t. Now, the hybrid, that is heterozygous, capital T small t, will give in male parent two types of gametes, capital T and small t, which we are numbered as 1 and 2. And in the same way, female will also show two types of gametes having allele capital T and allele small t, and they are numbered as 3 and 4. And when self pollination is allowed, then the male gamete 1 is able to fertilize female gamete 3 as well as female gamete 4. So it is written 1, 3, 1, 4. The combination 1, 3 will be capital T, whereas combination 1, 4 will be capital T and small t will be heterozygous. Again, then the second male gamete, which is small t, can fertilize the female gamete having capital T allele as well as female gamete having small t allele, and therefore the combinations will be 2, 3, that is capital T small t, and 2, 4, that is small t small t. And it was shown that F2 progeny showed tall is to dwarf in ratio 3 is to 1, and in F3 generation, some tall plants gave only tall, whereas two-thirds of the tall plant again segregated into tall and dwarf in 3 to 1 ratio, whereas the dwarf plants gave only dwarf. So the F3 progeny shows 7 is to 3, tall is to dwarf. Wherever there is capital T, it will be tall, and the tall can be homozygous as well as heterozygous. If it is homozygous, it is capital T, capital T. If heterozygous, it is capital T, small t. Now, this is the genetic expression of monohybrid cross by Punnett square. You can see the parents tall, male, and dwarf, female, and will form capital T small t gametes, and the hybrid will be capital T small t. And then in F1 F2 generation, there will be two type of male gametes and two type of female gametes, which will result into phenotypic expression 3 is to 1 and genotypic expression 1 is to 2 is to 1. 3 means tall and 1 is dwarf. 1 is pure tall, 2 is mixed tall, and 1 is dwarf. Now, Mendel's conclusion on the basis of monohybrid cross, something is transferred from one generation to other, which carries information for expression of the trait. This something is particulate in nature. Mendel called this particulate thing as factor or determinant. Mendelian factor is now termed as gene. Genes are present in linear fashion on specific loci or position on homologous chromosomes in pairs. Each gene exists in two forms or alleles and govern expression of alternative phenotype or allelomorph such as tall and dwarf. You can see in the next slide the how the chromosomes are present as homologous pairs and how genes are present on a specific locus. Now you can see capital T, small t are the two alleles of the gene T dash, which is responsible for the height of the plant, and it has two alternative forms, capital T and T dash alleles. Capital T is responsible for tallness, whereas small t is responsible for dwarfness. Tallness and dwarfness are allelomorphs. In the same way, for color of flower, the two alternative forms may be purple versus white. Then a gene may have two or more alternative forms, but only two of them are found within an individual or a cell at a time. On the basis of results of F1 generation of monohybrid crosses for all the 7 traits, Mendel proposed that in a cross pure for two contrasting or alternative forms of a trait, the one which expressed itself in F1 generation is dominant and is able to suppress the expression of other or recessive form. In F1 generation, all plants were tall. It was seen that in F1 generation, all plants were tall. On the basis of results of F2 generation, obtained in monohybrid cross, Mendel proposed his first law, that is law of segregation or law of purity of gametes or law of unit characters. This law states that when a pair of alleles, alternative forms of genes are brought together in a hybrid, they do not mix up or blend but remain associated with each other and separate or segregate from each other in next generation when hybrid forms gamete. Means they are not never mixed up, they remain as it is and then separate. So in F2 generation, both tall and dwarf plants were produced in 3:1 ratio. Now, this slide shows a cross between tall plant and a dwarf plant. And in F1, you can see all the plants are tall. In F2 generation, 3:1 ratio can be seen. Three plants are tall, only one is dwarf. Now this again shows a monohybrid cross between purple flower and white flower. F1 will give purple flower having capital P small p, which is heterozygous or hybrid, and in F2 generation it will give rise to 3:1 ratio purple is to white, that is the phenotypic ratio. But if we see the genotypic ratio, then only one P is only one purple plant is homozygous, whereas other two are heterozygous having capital P, small p, and white is again homozygous. So the ratio becomes 1:2:1. Here again we can see the genotype of purple flowers. The first one is homozygous, whereas the second two are heterozygous having capital P and small p, and the white flower is always homozygous having small p, small p. So the ratio becomes 3:1, and here it becomes 1:2:1. Three are purple, the fourth one is white. Three are purple, fourth one is white. So it is 3:1 is the phenotypic ratio, whereas 1:2:1 is the genotypic ratio. Now this again shows the cross between round and wrinkled, and in this two crosses are shown. In the left side, a round seeded plant is male and wrinkled seeded plant is female, and in the right side it is just vice versa. In both the crosses, the results are same. In F1, all plants are with round seeds. In F2, 3:1 phenotypic ratio. In F3 again, some of the round seeded plant gave only round, whereas some gave round and wrinkled in 3:1 ratio. Now, these are the data which were obtained by Mendel when he performed the 7 crosses, round seed, wrinkled seed. The data were 5474 were round seeded and 1850 were wrinkle seeded, and total was 7324, and the ratio was 2:96, 2.96 is to 1. In the same way, we can see the data of yellow seed and green seed, purple flower and white flower, inflated pod and constricted pod, green and yellow pod, axial and terminal flower, tall and female flower, and we are able to see that the ratio is not exactly 3:1. It is approaching 3:1. This is the same data showing cross between the seven traits in their alternative forms. Now, with this, I think I have been able to explain you what is Mendel's monohybrid cross and what are the laws based on this cross. Only one law is based on this, that is law of segregation or law of purity of gametes or law of unit characters, which states that in a cross between two plants pure for alternative forms characters, the one which is dominant appears in F1 generation, and in F2 generation the two alternative forms are segregated, or two alleles are segregated, and the ratio obtained is 3:1. So with this, the class has been completed, and let us end the class.